by Faculty of Marine Science and Fisheries Hasanuddin University. And we are very honored to have a distinguished speaker who will share valuable insight on marine science, particularly related to the fisheries and a deep sea ecosystem. Thank you very much for your willingness, Mehdi. Yes, to officially open this webinar, it's our great honor to invite the Dean of Faculty of Marine Science to deliver his opening remarks. To Professor Mahatma, the screen is yours.
Thank you, Widya, as moderator. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Good evening to Meti Rodigu. I hope I spell your name correctly. Okay, my name is Mahatma Lanuru, the Dean of the Faculty of Marine Science and Fisheries. On behalf of the Faculty of Marine Science and Fisheries, it is my great pleasure to welcome you all to our SymarFish webinar series. So this webinar is important milestone to leading up to the international and national symposium on marine science fisheries that will be conducted in June this year. Well, our webinar topic today is from fisheries to deep sea and everything in between. So this topic demonstrates the complexity of our ocean. The topic also reminds us that our words, whether it is supporting local livelihoods through sustainable fishing or exploring the mystery of the deep sea, it's far on interconnected a global system. Today, we are very lucky to have a special invited speaker, Meti Rodigu, who is the Chief Operating Officer of Ocean Alliance. With her extensive background in fisheries management and ocean technology, Meti will embody the spirit of our topic, bridging the gap between the traditional marine science and the future of the deep sea exploration. To our students, researchers, partners, and all of the participants, I encourage you to actively participate in this today's session. Let us use this time to be curious and work together as we prepare for the big symposium in June later on. Thank you for choosing to spend your time with us today. We hope that you'll find this webinar both enlightening and inspiring. And special for our speaker, Meti, thank you for joining us today. And thank you also for sharing your knowledge with us. Thank you. Please enjoy the webinar. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much for our Dean for the inspiring remarks. Now we are moved to the speaker. This is the very important one. But before that, it's my pleasure to introduce our speaker first. Let me to read your briefly CV. I will share the file first. So yeah, Mehdi, this is your executive profile. Mehdi graduated from conservation biology in Arizona State University and finished her graduate a dual master degree in marine biology and marine policy in University of Miami. And Mehdi is a session ocean professional with over 15 years of experience bridging scientific research, ocean policy, and technological innovation from shaping federal ocean policy in Washington, D.C. to directing global sea expedition. She is dedicated advocate for marine mammal protection, sustainable fisheries, and the deep sea as well. After as a science program director of Ocean X in 2019 until 2050. Right now, she is a Chief Operating Officer for Ocean Alliance, that organizational for strategy, technology development, and global fundraising for 50-year-old whale conservation for a nonprofit organizational. So that's the very brief executive profile for Medi Rodrigo, our speaker today. And yeah, today she will deliver a presentation entitled From Fisheries to the Deep Sea and Everything in Between. It's a very catching topic for today's holiday, actually. And without further ado, Maddie, the screens absolutely is yours.
Okay, can you all see that? Is that coming through okay?
Yeah, that's clear.
Okay, perfect. Okay, hi everybody. My name is Maddie Rodrigue. I am coming to you live from New York City. So apologies if you can hear any traffic outside the window, but I know from spending so much time in Indonesia that traffic is very much a part of life. So I wanted to talk to you all today for part of this seminar series. Because I'm at a little bit of an interesting time in my life where I'm finishing a chapter in my career and moving on to the next. And so this talk, From Fisheries to the Deep Sea and Everything in Between, is going to focus a lot on my research. But the core theme that I want you all to take away from this, and we'll circle back to it at the end, is really allowing curiosity to drive the different chapters in your own careers and really to drive your research. Because without curiosity, we don't generate new knowledge. We don't explore. Without that new knowledge, we can't protect and conserve the spaces that we deeply care about. The agenda for the talk today is first to just tell you a little bit about myself, because each of the components of my biography also correspond to different chapters of my own research and, again, my own curiosity. So the first chapter of my career really focused on fisheries. The second chapter of my career really focused on the deep sea. And through those two chapters, I really started learning about everything in between, everything that connects the deep and the shallow environments, and everything that really inspires people to care about the ocean, but also the sort of criticality of the ocean to coastal communities, and the interactions between humans and the environment through fisheries, through the deep ocean, and now in my newest chapter through marine mammal conservation and megafauna. That's me in the sub, if you can't tell. That's me at about 800 meters underwater. Okay, so I started my graduate work at the University of Maine, and I was studying fisheries science, so a graduate degree, a master's in marine biology, and a master's in marine policy. And what I really was focusing on was looking at coastal communities, looking at resilience, looking at their dependence on fishing, especially what's called the groundfish fishery in the US, which is finfish, flatfish, like this big one that you can see on the screen, and working directly with commercial fishermen to try to implement sustainable fishing practices that then they could carry into future fishing efforts. After I moved on from graduate school, I started a fellowship at the National Oceanic and Atmospheric Administration. You're all probably very familiar with NOAA. It's the U.S. agency that does all ocean weather and climate science and policy. And so through that work, I started learning a lot more about deep sea ocean exploration, but also about international governments, international policy, international communities, and how more international communities and audiences really perceive the ocean, and in a lot of ways depend on the ocean for their livelihood. So that really started to expand my curiosity and expand my scope. And it was there that I discovered a passion for ocean technology and for the deep sea. And so it felt like a pretty hard pivot at the time, but you'll see how it connects later. I got really interested in the deep sea, and after my fellowship at NOAA was over, I found an organization called OceanX. OceanX is a nonprofit. I'll go in and talk about it a lot later. Some of you are probably familiar with OceanX. I know Ijay is because we got to sail together in 2024 on an Indonesian expedition. But OceanX is really dedicated to studying deep ocean environments as well as shallow coastal ecosystems, but mostly deep ocean environments, and really trying to explore, discover, and bring that knowledge back to the world through storytelling, through media, and try to change hearts and minds to really care about advocating and protecting the ocean. Now, I've just recently taken her after 7 years with OceanX. I built the program from scratch. And so after 7 years, spending most of my time on the ship that you saw in that photo, and building the scientific program, I decided to transition away from OceanX and spend a little bit more time on land, and focus on a suite of species and a topic that is near and dear to my heart, and that's marine mammals. And so I accepted a role with another nonprofit called Ocean Alliance that's based in New England. And what they really do is look at innovative ways of studying and conserving whale populations around the world. You can see here's a little drone called the SnotBot, which is basically a drone that flies through the whale snot and collects a sample on Petri dishes that can be analyzed for hormonal analyses. You can look at all kinds of things to check the health of the whale non-invasively. So that's just one of the tools that they utilize. And so I'm really excited to begin that next chapter in my journey as well. So where I started, and this is very, very fitting for a fisheries program, and so you probably already all know this, but please bear with me. For fisheries and my study in fisheries, I was based in New England in the state of Maine, and so that's the one that kind of looks like a mitten on the right hand that I circled on the screen, and on the graphic of the United States on the left, or your right, depending on which way you're looking. And I was really responsible for what are called the New England groundfish stock. So most of you probably know that the way that we measure fisheries in the US, fish stocks in the US, is to look at things sort of species by species. We try to estimate how many fish are in the sea, basically. And that's really hard because you can't see them. You don't know where they're going. You don't know how long they live. There's very, very limited data. So we try to collect what are called biological reference points to input into stock assessment, which basically can then set advice to the managers of the fisheries to let them know, okay, you can take this many fish out this year and there will still be enough of the population left over to reproduce, and to make sure that we can continue to sustain not just people, but also sustain the populations of fish. And so as you can imagine, this is really difficult to do. Counting fish is really, really hard. It's very, very difficult to sort of find the populations, to understand how they're changing, in part because these surveys, they're trying to mimic all of the commercial style fisheries in the United States, but often they're only using one gear type, which is the gear type of the bottom trawl. So often you have species that don't even interact with bottom habitats, with seabed habitats. You have species that are in the water column, but really the only gear types that are available to the surveys, the research surveys through the government, are sort of very similar types of gear that is mimicking a commercial fishery. So to supplement that, they try to incorporate information from the actual commercial fisheries, which sometimes can be biased, sometimes not, depending on the trust between the fishermen or the fishers and the government officials in those regions. Every region in the US is different, and I know for a fact it's the same in Indonesia. After spending time studying fisheries there, it's infinitely more complicated than this. So please, as I said, bear with me. But the other thing is that it's really expensive. You have to get the ship, you have to pay for the fuel, you have to pay for the time at sea. And we all know that can be really, really expensive. And so these surveys are not done at a pace and a scale that matches the life history strategies of the species that they're trying to measure. So in some cases, you have fish that are really quickly reproducing, and sometimes you have things like what I was studying, which can live 60, 70 years and don't become sexually mature until, they're 20. And so it's really, really hard to basically spend the money and allocate the funding to measuring these in a way that really matches the life history strategy. That is also really difficult because you have all these limitations around catchability. You have all these limitations around gear types and sort of seasonality. But we know that the ecosystems are changing now more rapidly than we can even keep up with because of climate change. And so all of these changes are really going to impact these fish and really impact these fish species. And so this was the challenge that I was presented with when I was in graduate school doing my fisheries research, both on the science side and the policy side. How can we design a way that is the right scale and the right gear type and the right seasonality, matches the habitat complexity of the seafloor, and also incorporates fishermen's local ecological knowledge. So the knowledge that they have just going out for generations and spending their lives fishing, where the fish are, how things are changing. How can we incorporate all of those into a stock assessment that can supplement the federal assessments that were basically missing the entire region and missing the complexity of the species in that area? And so what we decided to do was conduct a longline survey. So a demersal longline survey. It's so funny because this is anybody that studies fisheries or goes fishing knows that this is a very typical gear type, right? But for some reason, it is very, very rarely incorporated. Well, for good reasons. It's very, very rarely incorporated into actual official standardized stock assessment surveys. Part of that is because it's a lot more variable than a typical trawl. The trawl goes in, it's like a scooping up all the M&Ms, and then the trawl comes out, right? But the long line is more complicated. The hook sizes matter, the soak time matters, pretty much everything matters. But the nice thing is that this also accounts for the habitat complexity. So it's not just sort of going to bang against the rocks on the seafloor or even destroy seafloor around it because you're just laying the long line down on the ground. This is really important because we were matching the life history strategies of fish, not just like that big halibut that you saw on the previous page, which I just like to include in slides because I feel like I'm bragging because it was so big. But species like the Atlantic cod, which you can see in my hands in the photo in this slide. And this is really important. This is really a finfish that built the northeastern United States. Like the entire northeastern seaboard was built around this little fish. And they were fished basically to extinction. And so what I was tasked with doing was incorporating all of these challenges into a type of survey that incorporated fishermen's knowledge, like these guys that you can see on the right. These are what local fishers look like in New England. And myself included, and really design something that could actually try to really accurately and sort of more transparently monitor these groundfish species. And so we were able to build this survey. It ended up running for about 10 years. We have a lot of data. There's a lot of reports and studies that are published out of it. But really, ultimately, what this taught me was looking at an environment, looking at an ecosystem in a different way, like trying to tackle these challenges, these questions we have about an environment that we can't see. And I'll add that when I was doing this work, I had no idea that there was a whole field of developing sensors and equipment and things that are specifically designed to go down and look around in the ocean. I didn't know that remotely operated vehicles existed. We didn't have access to things like drop cameras. The best we had was, a GoPro that we could stick on the line. And then once it gets too dark, you can't really do anything about that. So it was very, very technologically limited. But this was the best way that we could innovate around what had been done for maybe 5 decades up until that point, to try to really supplement that information. And we had a lot of really major wins in terms of incorporating this monitoring data into the actual federal stock assessment for the Northeast United States. So I got really interested in policy and I got really interested in working with coastal communities. And so I decided to take on a policy fellowship at NOAA. And so you'll see now all of the slides that are NOAA affiliated have the NOAA logo on the corner. Thank you, NOAA. And so I started working for NOAA Research, specifically for the chief scientist of NOAA. So setting the national research priority for the United States. This was way a much bigger job than I had ever thought that I would do ever in my life. It was a huge honor to learn how to look at all of the ongoing ocean, weather, and climate labs, cooperative institutes, programs, universities, and strategize where the gaps were and try to develop and build national ocean policy that then would then allow programs to allocate time and funding and capacity to start getting at some of these things that we desperately needed to understand. One of the things that I didn't really think a lot about because I was so fisheries focused was how massive the ocean actually is. I was just looking at my little slice of New England at that time. I was so dedicated to my research and my graduate work. And, the farthest out I would think of would be, the Gulf of Maine. And those were the communities I was working with. When I started working at NOAA, I started thinking about the ocean globally. And what I learned was absolutely astonishing to me. At that time, probably 15% of the global ocean had been mapped. This was in 2018. Now we're up to about 28%, and that's largely through efforts like Seabed 2030, which is the UN Decade of Ocean Science initiative to map the global ocean by 2030. That initiative actually started while I was at NOAA through my boss at the time, the chief scientist of NOAA, and the UN Decade Initiative we were able to work on and sort of organize in the beginning as well. And this basically came from an exercise that we did. This is the behind the scenes look, an exercise that we did in the US where we were looking at how much of the US had been mapped. And the answer shocked us. Nobody had an answer. Everybody was estimating, everybody was going back through old survey data. And these are like, like US official research programs. I was so shocked. And the reason is because nobody really understood why mapping was important. Nobody really understood. There was no case for the deep sea. And at that time, there was a really, really huge limitation in the technology and the expertise that the US had, and also the globe had access to. Some countries had access to deep sea technology and equipment, especially if they were really focused in oil and gas or offshore wind or industries like that. But most of the organizations and most of the other government agencies that I spoke to had a huge limitation in access to some of this technology that allowed them to shine a light on the deep sea and really allowed everybody globally to understand like what a huge challenge this was going to be to actually try to characterize and map our planet. Not only that, it's characterization, right? So 28% at this point has been mapped, which is amazing, but like less than 4% has actually been visually observed. And that's also, again, due to these limitations in tech and expertise, in access, in equity, in sort of even commercial data being shared with academia, being shared with, US or global nonprofits or the UN Decade of Ocean Science and initiatives like Seabed 2030. So all of these topics were started percolating and swirling around me, and I was very fortunate to be able to work on the U.S. National Ocean Policy in 2018. And in a sense, the policy was, you shall go map the U.S. exclusive economic zone. And I put it forward, and the White House accepted it. So that was the policy from there on out. They said, we are now, as a nation, the United States will focus on the deep sea, will focus on mapping the U.S. exclusive economic zone because we can see the benefits to our U.S. blue economy. And also we would like to make sure that we're leading the way in all of this. So again, a huge honor. But before this year, I didn't even realize this was a problem. I didn't even realize this was a challenge. And I got so curious about all of this, and all of these things really started swirling around. And I started wondering about access and innovation and why we were so behind on all of this, and how people across the world don't even think about the deep sea at all when they think about the ocean. And so then I found an organization called OceanX, and they were very much just starting out at the time. Prior to their global research programming and their ocean going operations, they were a production company. They did films like Blue Planet 2. They had an incredible impact on the world. They were operating on a much smaller vessel, and they were doing a lot of filmmaking. And it was very much like they wanted to show and tell the story of the ocean and try to get people to care. The founder, Mr. Ray Dalio, and his son, Mark Dalio, decided that they wanted to actually equip scientists with the right tools to be able to get at these big questions, these big challenges. What's in the deep sea? Why should we care? What does this mean to us? And so they decided to build the Ocean Explorer, which is the vessel that you see here. And the whole goal of OceanX is this is a nonprofit, but it's dedicated to exploring the world's oceans and bringing discovery and science back to the public through captivating media. The way that we did that was basically to facilitate science and scientific research through underserved nations. So basically nations that do not already have access to this type of technology, which I have to say is pretty much every country in the world. This is a vessel that has multi-beam echo sounders and sonars to map the seafloor. It has sonars to map underneath the seafloor. It has 2 submersibles that can take scientists down into the environment. It has a 6,000 meter rated ROV. There's a helicopter for aerial surveys. There's labs on board, environmental DNA sequencing equipment on board, shallow water mapping capabilities, a full dive center. I could go on and on. I designed it, worked there for 7 years, built the program. So it was very multidisciplinary and very comprehensive. And you can ask Widya about her time on board as well. And so we would bring the vessel to these countries that would ask us to come, and we would say, okay, what science do you want to do? And I would work with scientists to build proposals and work on projects. And through that, I had to become a quasi-expert in every single thing that they wanted to do. I had to understand enough of the biogeochemical oceanographic principles around their survey design to know that we have to build these CTD casts in this way. It has to be supplemented with the multi-beam sonar or the EK80, the fishery sonar in this way. And oh, by the way, did you know that we have environmental DNA sequencing on board? Do you want to layer in the characterization of the environment biologically while we're collecting all of those information and create a full picture of the environment? So that was my job, but that was the work that I did with the researchers on board. And it was built in a way that it would also tell the story of that science. So in addition to being a floating research institution, it is also a media production center. And every component on the vessel is designed to look really cool on camera, but also be really, really functional. So it was critical. So during my time at OceanX, I took the vessel on scientific expeditions in every ocean basin. We mapped over 1 million square kilometers, all for the most part, where we were permitted by different countries, made publicly available, so all scientists can look and learn. And through that time, we published studies, probably like 140 scientific publications in those 7 years. You can find them on oceanx.org. I didn't want to just give you a block of text with all of the words and the links and everything, but they're really interesting because you can really see how multidisciplinary the vessel is, and the studies that I got to work on for my research were everything from deep sea ecology, microbiology, fisheries, MPAs, marine mammals. I could go on. In particular, the ship was very unique because it gave me a platform to really innovate. It gave me a platform to really test out the limits or the limitlessness of my curiosity, because everything that we dreamt up, we could do, we could design, we could build. And through this, I got really interested in particular of these very specific geomorphological features on the seafloor called seamounts. And this is where my fisheries background really came into combination with my deep sea background, because these seamounts were super interesting. The seamounts themselves are really interesting because they are like hotspots for biodiversity. And that's why you always see fishermen going out to these seamounts and fishing around these hotspots. The fishermen know, even if we don't, right? Even if we haven't mapped it, the fishermen know where they are. So I've always been really interested in seamounts and actually getting to look at them and map them and survey them. I started thinking about ways that we could actually bring them to life. How could we characterize them? How could we advocate for their importance? And the opportunity really came to design a study with our partners through the Around Africa expedition in 2025. So this was a massive expedition. I have a map of the whole ship track that you can see. I sailed on this entire expedition. It was like 4 months long. And along the route, we were collecting oceanographic data. We were looking at eDNA. We were helping NASA ground truth their PACE satellite. So doing lots and lots of different ocean observations, ocean observing, oceanographic observations, etc. Everywhere where you see a triangle is where we had a dedicated mission. And these missions were, these expeditions were really focused around seamounts. And one that was really exciting to me was when we went to Cabo Verde, Cape Verde. So on the circle there off of Africa, and then a little bit bigger, you can kind of see some of the partner logos. This was called our Digital Deep Expedition. And this is in partnership with a group called Ocean Quest, with the UN Decade, and with IMAR, the Institute for Marine Research in Cabo Verde, with the local scientists there. And so you'll see their little logo down here. I have to credit my dear friend Yara Rodriguez for all of the work on these slides for and with me. So there are seamounts that essentially make up Cabo Verde. So Cabo Verde is a chain of islands. They're all volcanic. And then there's all of these seamounts, about 10 of them underwater. And really you can see how rich they are just by going out and seeing all the fishing activity. They're really important for other things as well. They're really important for climate regulation. They're really important for all levels of trophic support. They're really important for different levels of biogeochemical cycling. And that's because seamounts are really interesting. They have these characteristics where because of the way that they raise up from the seafloor, sometimes underwater mountains, like underwater landscapes, like Everest underwater, right? And so you can see that they are changing the patterns of the currents around them. And there's an effect called upwelling that happens. Basically, colder, nutrient-rich water is driven up the side of these seamounts, and they create these hot spots of food for big animals to come in, small animals to come in. And so everybody was talking about this in an oceanographic context, because this is obviously really important for fish and fisheries. What I was thinking about was the essential fish habitat, basically nursery grounds for all of the fish to go and have baby fish for them to grow up and to actually then become part of the stock complex and accessed by the fishermen and support sustainable fisheries. And so we decided to design a study around this concept, around these critical habitats that allow other animals to thrive and support other ecosystems. And these are called vulnerable marine ecosystems. And so we were looking for specific species that actually made up what was called a vulnerable marine ecosystem. And this is a FAO defined term. And basically, the FAO and the UN came out under the UN Convention of the Law of the Sea, came out and said, if you find a habitat that can be classified as a VME, a vulnerable marine ecosystem, that area needs to be protected, because this area will help support all of the other areas around. I'm paraphrasing, but this area will help support all of the areas around it. And so the qualifications for something to be classified as a VME are essentially having these different types of species of coral and sponges that create these nursery grounds, that create carbon storage, that create this complex habitat structure for fish that then are recruited into a fishing stock. That's really important, obviously, for the sustainability long-term of the stock. Not just the fact that these are hotspots that create these nutrient upwellings that then all these other megafauna come in and eat the little things and the big things, and so it goes. Okay, so we decided that we were going to do an expedition focusing on one seamount complex in particular, and this was called the NOLA Seamount Complex. And I loved it because it's a twin seamount complex. I am a twin. But it also was really cool because we actually got to compare the differences between these dual peaks. So this is actual mapping data that you can see on the screen here. This is what the Ocean Explorer collected. So we surveyed the area, and then we basically wanted to design a layered multidisciplinary study to get at what the seamounts were made of, what lived on the seamounts, what we were visually surveying for our underwater visual census, which is basically standardized transect lines for the ROV and the submersibles at the same time, so we're covering more ground. What the oceanographic parameters of those seamounts were, so using the CTD and the rosette to look at all of the different oceanographic parameters, as well as sample for environmental DNA for all of the animals that are too camera shy and we can't see them. And so we need to sample DNA around their environment and try to see if we can pick up the molecular signal in the water. And then collecting different specimens that can be used that we think are new or can be classified as some of those VME specimen that then basically we worked with IMAR to create a genetic reference base for. Okay, so that's a lot. We're also doing bongo nuts. We're looking at ichthyoplankton and zooplankton, obviously very important. It was a big study. And what we ended up finding was really interesting. So we were comparing the different seamounts as well as the different flanks, and we're looking at different benthic communities depending on the depth strata. So what you can see are on the seamounts, the white line are the different contours of the seamount. And again, I'm sorry, you guys probably already know this, so just bear with me. But the contours are the white line is the contours, and then these are just an example of some of the transects that we did. Okay, so the purple is the ROV, the yellow and orange are the two submersibles working together. And what they're doing is a standardized visual survey. So they're keeping the same distance from the seamounts. The cameras are all configured to have the same fixed focal area and fixed focal length. Lighting is the same. And you basically just have like 20 scientists like frantically typing as the video is coming in and trying to classify all these organisms. And I'll get back to that in a minute. And so we're looking at these distinct benthic communities and we're identifying VME taxa at different depth strata. This is really, really important. This tells us that at every level, this ecosystem is sustaining life in addition to the life that we're observing. This tells us that that there is connection between the deep and the shallow. This tells us that it's not just what we can see that's important to protect. It's not just that what we can sort of, throw with a little lead weight down and know that we hit the bottom of. It's also into the depths. There's life there, and it's really important. We found VME taxa like glass sponges and soft corals. We found black corals, whip corals, and even extremely, extremely dense black coral coverage, really old, really critical for supporting fish in their juvenile stages, for essential habitat. And we found that in extremely high magnitudes. And so all of this was packaged together essentially to give to the managers in Cape Verde to make a case for protection of these seamounts, or at least creating a sustainable mixed-use fishing plan that still allowed the fishermen to access these areas that were critical for their survival, but also basically you're finally visualizing the habitat. You know what gear type you should or should not be using. You know what lives there. You can see it. You can design a fisheries survey to monitor using either molecular non-invasive tools or use tools that actually align with the type of habitat and the type of fish species that you see. So this is a huge development for our Cape Verde, but also for people like me who start with fisheries, go into the deep sea, and this is the connection between those two. This is obviously a lot of data, so I just wanted to mention quickly that we developed a tool that was really cool to analyze all of these data. It was like 40 terabytes of data, something crazy. And that's because most of it was visual, and then there was also molecular. And so we developed an application that the scientists could use instead of that frantic typing. They would be able to look at all of the images that were collected by the ROV and the subs after the dive, and then go through and basically swipe through the different species and train an AI model that already had been fed some baseline data. They were training an AI model to basically say, okay, here's what's here, here's what's not. Out of that AI model came what's called predictive biodiversity tools. So what we do is we can say, okay, this is the transect line along the side of the seamount, and along this transect line, here's the presence and absence, or the relative density or relative abundance of these really critical VME taxa. You can look at the habitat and substrate type from the mapping data, ingest that into the tool, and then sort of extrapolate what you think you might see across the rest of those habitats, across the rest of the seamount. And this is really important because, again, deep sea exploration is super expensive. It's very time consuming, and you need to really be developing these types of tools so you can pull this up to managers and say, okay, we know that this is what we found in these specific areas. Based on this data, we're going to interpolate that across these seamounts. This is what you're going to find at different depths, and thus make an even stronger and more compelling case for protection or sustainable mixed use planning. out of this predictive biodiversity tool also comes something that I'm still working on with OceanX actually, which is creating a digital model of these seamounts everywhere we go. So being able to sort of pull up a 3D visualization of the seamount, pull up a dive and actually go on the dive and look at the classifications in real time. And obviously this is all driven by real data, but this is also going to be a really powerful outreach tool. So participants can go on the dive. It's really important for scientists, extremely important for our managers, policymakers. You can't bring everybody out there with you on the boat, on the ship. So you might as well develop a tool that can make them feel like they're there and really can see where these hotspots are. I basically just said all of this, but essentially you get maps that allow you to that can tell you where things are, where things aren't. And then if you have certain environmental parameters like slope angle, substrate type, even things like temperature, et cetera, you can basically ask the model, if the temperature increases by 1 degree over the next 20 years, what happens to the black coral? And you can really start to see how the distribution can change. And again, this is all models. And my favorite fisheries advisor was a population dynamics modeler. And he was building models all the time. And he said his favorite line is, all models are wrong, but some are really useful. So obviously this is only as good as the data you feed into it and how you train the AI model, et cetera, but it can be very, very useful, not just in understanding what's in a deep sea environment, but also being able to tell the story of that deep sea environment and plan future research. So you can save time, you can save money, and you can really target where you need to go to get the data that you think is going to best serve your study. Okay, so like I said, after OceanX, I took 6 months off. I was spending probably 3/4 of the year offshore. I was running the science program from my office on the ship. I decided essentially after 7 years to take a step back. They're still working over there. They're doing an amazing job. But I decided with my next chapter, I was going to focus on leading a company into the next sort of iteration of their legacy. And so this is Ocean Alliance. I really encourage you guys to take a look online about what they do. They use innovative tools like drones to really understand whale populations, their environments. They also really prioritize education and outreach. And I am really, really excited to be part of their team. They've developed things like Snotbot, which I mentioned at the beginning. Definitely check them out, whale.org. Shameless plug for my new organization. And that brings me to the end of my talk. And so this is the biggest takeaway, I think, out of the entire talk. As I mentioned, the research elements are really important. You guys are at university, you think about research all day long. When I was a student, it was really hard for me to pull my head out of my research and really think about some of the big picture things and really understand how I was thinking about the world around me and my place in it. Now that I'm only slightly older than I was as a student, I've really decided that the most important things to me are letting my curiosity drive where my career goes. What am I curious about? What do I want to know? What drives me? What pulls me? Because I found, as I've given you examples, that curiosity will drive innovation. It will drive you to think about ecosystems in new ways, to think about our understanding of the ocean in new ways, to really use that innovation then to drive discovery, to make these discoveries about, it could be something very small, it could be something paradigm changing, but without actually using that curiosity, driving innovation, going towards that discovery, making that discovery, we really can't generate new knowledge. This looks like scientific papers. This looks like storytelling. This looks like things that basically humans were not aware of. And now we are collectively as a species aware of. And this is really, really important. That knowledge then drives protection, whether it's through policy, whether it's through shared use planning. And this is a cycle, right? Because there's always going to be something new. We don't know anything about this planet. And the last thing that I'll say that I have really, really, really always identified with is that outreach for all of this, outreach for scientific work, outreach for the generation of knowledge is not just sort of the last step. It's not the afterthought. It's not that, okay, I have published this paper, I have defended my dissertation, and now I need to go tell people why it's important. Outreach should be at every single step of this cycle. Tell people why you're curious, tell people what you're thinking, tell people what you're innovating, what you're designing, talk about your discoveries, talk about the knowledge that you're generating, and get people to care so that if people can come with you along for the journey that you're on, they'll be more inspired to also join you in the quest to protect our ocean for a better shared planet. So with that, I would say terima kasih. Thank you very much. I really appreciate the time. I hope I didn't talk too fast. I have a tendency to do that. And any questions, I'm very happy to take them.
Okay, thank you so much, Mehdi, for your insightful and inspiring presentation. And your presentation gives me like a flashback. Yes, flashbacks go to during the 2024 mission, and when I had a chance to use the Mars submersible before descending beyond 200 meters deep, and I thought I would probably only see large fish like marine mammals, but yeah, I was wrong. In addition of sharks, I also observed colorful deep sea corals. I mean, like sponges and many small fish and crustaceans. That what I want to highlight is that the deep sea is actually very rich and diverse ecosystem, comparable to shallow water ecosystem that we really easy to see surrounding us. Okay. Now we are welcome the question from the participants. You may raise your hand or type your question on the chat box. We still have like 10 minutes for question and answer. And yeah, before you ask the question, please briefly introduce yourself. Okay, any question from the participants?
Boleh,
Okay.
Wit?
Yes. Yes, go
Okay.
ahead, Bu Yayu.
Okay, thank you for the chance. Good morning, everybody. Sorry, I could not find the present question, so I just okay. Thank you. Thank you, Mati, for the very interesting presentation. And I saw that from your. Sorry, I was late, but what I could see was that you survey also the seamounts of the Africans area, and I was wondering if it looks so high biodiversity, of course, and it's so stunning to see that. But I was wondering if, based on your experience, how would you compare that to the area, the seamounts of Indonesia? Did you see any maybe different? They are the same or slightly different or something like that? And also, I was wondering if you have done also surveys on different time of the year, like different seasons. So, did you find also any seasonal effect on your survey or something? Because it's very interesting to see that to know that also if there's like if you survey on different times of the year or different season, you might find any seasonal effect or different results on your survey. So that's all. Thank you. Thank you, Vidya.
Thank you for the question. Matthew, would you like to respond?
Sure. That's a great question. And yeah, as a fisheries scientist by training, I'm always thinking about seasonality when I design a survey. One thing with the the seamount survey, we weren't actually targeting any sort of mobile organisms, okay? So we were only targeting, for the visual survey, we were only targeting things like sponges and coral that aren't moving. For any of the other environmental parameters, absolutely, I would expect to see seasonality. I would expect all of the different oceanographic dynamics to change depending on what time of year we were there. But I wouldn't necessarily expect the presence or absence of the sponge and coral taxa that we are looking for to characterize the environment as a vulnerable marine ecosystem to change all that much. For the predictive tool, where we're looking at predictive biodiversity, again, we're just looking at things that aren't moving around. But if we wanted to try to develop a tool for that, for like fish and fisheries and for megafauna, for example, and even for plankton or ichthyoplankton, we would absolutely have to go back and survey much longer for many more times during the year to try to get at that seasonality element. And your first question was about the comparisons between Indonesia seamounts and African seamounts. When we were on the Indian Ocean side of Africa, there were a lot of similar species composition as when we were on the Indian Ocean side of Indonesia. So while a lot of the sort of fish species were not quite the same, a lot of the, again, the organisms like coral and sponges, I like to say that I saw some familiar friends at both. And so that was really interesting. The oceanographic dynamics, though, were quite different. And so we're still looking at those results and looking at those studies. And that's also something that a lot of the Indonesian researchers are looking at as well. And what actually, this is a shameless plug, but what came out of that expedition was there's a group called Challenger 150, and they look at all of the seamounts across the world, and they try to basically make an advocacy case for seamounts and for visual census. And so out of the 2024 Indonesia mission that OceanX did with Indonesian scientists, a new working group of that Challenger 150 was established for the first time, led by Indonesian researchers and researchers from Malaysia as well, joined by African researchers and Seychelles researchers, all about Indian Ocean seamounts and looking at the dynamics. The last thing that I'll say is that only in Indonesia did I see a coelacanth. So that basically won everything for me.
Okay. The silicon is was like extinct, yeah. The fish,
They thought it's alive. Yeah, it's not extinct. Yeah.
So that's but then I forgot the year when they actually found it also in the north of Sulawesi, where they found it in the fish market.
Yes,
So
exactly.
it's interesting to know that it's still there. Yeah.
I almost included a picture of one that we took from when we were in Sulawesi, but I didn't have time or enough slides, but I should have.
Yeah, thank you.
Okay, before we move to the another question for all of the participants, please feel free to fill the attendance list. So after this, you will have your e-certificate and material for this webinar. Okay, we have Andi Alia here that want to ask a question, please. You can unmute yourself, Alia.
Okay, thank you, Ibu Widya, and also thank you for the presenter. Thank you for the insightful presentation today, and it was truly wonderful sharing from you. So, hello, my name is Andi Alia Yusria. You can call me Alia. I'm from Department of Marine Science in Unhas. So, basically, I'm focused on marine microbiology. So it's all about microorganisms. So I just wondering, like, basically in methodology, like the methods, I believe that most of you guys on board, you will conduct a research also in microorganism. But I would like to know how you culture it and how you keep them on board, because there are a big difference in environmental between surface microorganism on surface ocean of the ocean, and also the deep sea microorganism, because uh, there is such a very big difference in ecology, such as high pressure and also low temperature. Maybe that's my question. Thank you.
That's a great question. So we use a variety of different methods on board. Uh, we basically any of the surface samples or anything that we can collect remotely with just the CTD rosette and the Niskins. We'll use the CTD rosette and the Niskins. But for specifically looking at like in situ sampling, to combine with the visual census, so we can basically say, okay, here's what's in our environmental DNA sample, and we can match it up to the video. We actually used a automated eDNA sampler on the ROV, and this was something that we got early in 2024. This is one of those really cool innovations that I worked with a team out of MIT basically to develop. And this was called the pufferfish. And essentially it has triplicate filters that you can put like 0.2 or 0.4 micron pore size, just like the normal standard cellulose ester or glass filters, the 45 millimeter diameter. And we can basically filter water as the ROV is flying. So you're getting that sample at depth. It injects RNA later into the sample as a preservative. So you can finish the survey. We compared the methods with and without the RNA later. It makes a huge difference. We didn't always have the RNA later working. So for some of the samples, we had to subset them out. But essentially we collect the water samples through those different ways. Either we filter them in one of the laboratories, or we would use that automated pufferfish filtration system. And basically, unless we were doing some sequencing on board, which we would use Oxford Nanopore, Prometheon or GridION sequencer, we wanted to try to keep everything with Illumina for this particular study, so we could compare it to the rest of the track where we were using Illumina sequencing. So in that case, we would preserve the samples in the RNA later in the -80, and then we sent the samples to a lab basically to get sequenced. I hope that answers your question.
Oh, okay, thank you. Because yeah, it's such a tricky thing, but you have such a very wonderful technology on board, so I believe that you can conduct that research.
Thank you. Yeah, we can't culture anything, which is a shame, but we can definitely collect the molecular samples and preserve them.
Okay, thank you.
Yeah, we have a few minutes for the question and answer. Is there any question from the participants? Yeah, one question from me. It's really a general one, but considering that a large portion of the deep sea remains unmapped and unexplored that you mentioned previously, in your site, what are the most promising technologies or approach to accelerate the deep sea exploration and improve our understanding of these ecosystems? Because I remember that I have read one sentences that doing a research in the deep sea is more harder than in the moon. Yeah. I remember that.
I can't vouch for the moon, but I can certainly vouch for the deep sea. It's hard. That's a great question. I get asked that a lot, and I'm kind of, I have my practical answer and then I have my emotional answer. So I'll give you the practical one with my science brain first. I really think that autonomous systems, robotics, deep sea robots that can cover really large areas and collect really high resolution photogrammetry and also mapping information are the way forward. I think the more that these are developed and built, the more cost effective that they'll become. But even things like inexpensive drop cameras to spot check certain areas, I think is really important. I think that molecular tools are definitely the way forward in terms of characterizing biodiversity. We have an incredible global ocean observing program through the Deep Argo and the Argo networks. But I do think that the future will be incorporating environmental DNA samplers onto those, or automated samplers onto those, as well as onto these underwater robots. So that's the practical that I think we can get the most amount of data, the most cost effectively, and really try to seek to understand our planet a little bit better. I personally, because I've dove in submersibles, and because I've witnessed how paradigm changing that is for our researchers, I really think that the data is one thing, but a true understanding of the ecosystem, of the environment, of the planet really comes when you can immerse humans into the environment. You can do that in better, more like pragmatic ways with immersive technology, headsets, 360 films, whatever. But actually getting into the ocean, getting into the deep sea is, I think, going to be what is the most paradigm changing thing to get people to really understand and care about the planet. It's just you can't bring everybody all the time, right? So having those tools that can really allow you to experience what it's like to dive in a sub, like by just putting on a headset or going to an IMAX film or going into a 360 dome, I think are going to be really, really important going forward.
Right. That's a very engaging discussion, by the way. Yeah, because the limit of time, Mehdi, thank you for this very good webinar that you share with us for your knowledge. We truly appreciate all the insightful question and response shared in this session. Mehdi and all the participants, before we close, I would like to invite all of you to join the International Symposium on the Marine and Fisheries that will be held on June 20, 2026. It will be a great opportunity to connect with researchers and professionals from around the world. Maybe we also welcome you if you have a chance to join us in Indonesia. Yep. So we warmly encourage you to register if you have a chance and be a part of it. Would like to extend our sincere gratitude to our speaker, Mehdi, and of course to our Dean for his opening remarks, and of course to all of the participants for your participation in this session. Okay, before we close, also we want to do a group photo, the online photo. Yes. Yes, if you're confident, please open your camera so we can have a group photo for this session. Yeah, for committee, can you please have a screenshot for? Yeah. So I will count 1, 2, 3 and smile. One more time. 1, 2, 3 and smile. Okay, with that, we conclude today's webinar. We hope to see you again in our next session. Thank you again, Mehdi, for your time. We appreciate that. And good evening. Have a good rest for you. And happy holiday to everyone. Wassalamualaikum warahmatullahi wabarakatuh. And have a wonderful day.
Waalaikumsalam. Thank you, Mehdi.
Waalaikumsalam
Thank you,
warahmatullahi
Widya.
wabarakatuh.
Waalaikumsalam Widya, mantap.
Jangan lupa isi absen.